So it's used to fertilize, say, the alfalfa fields. Okay, and here's exactly what you're talking about. So these antibiotic resistant bacteria get sprayed on the plants, the alfalfa plants. They die, right, because a lot of them are that resistant. But then what happens is the alfalfa then becomes contaminated with those antibiotic resistance genes, the DNA, just exactly what you said, contaminated with the DNA, some of which has those antibiotic resistance genes. And the weird thing is, you guys are thinking maybe DNA can remain intact longer in the environment than we thought. Okay, so then now you've got alfalfa plus these antibiotic resistance genes. It gets harvested. Okay, so we make hay out of it. We make alfalfa hay out of it. And where does the alfalfa hay go to? Back to the cows, right? So what they're trying to do with farmers is they're trying to get them to well, first of all, reduce levels of antibiotics. And then they're also trying to make farmers more aware that they have to be careful with what they do with the manure, because they might be like recycling the antibiotic resistance genes. And just like you said, why is this a big deal? Because when the cows eat the hay, if those little fragments of DNA carrying antibiotic resistance genes make it into the rumen and the intestine, just like what you said, through transformation, the sensitive bacteria in the digestive tract of the cow could take up those antibiotic resistance genes. So it's like you're feeding antibiotic resistance genes to the cattle. They're sensitive bacteria being transformed to them, and now voila, we even have, more generations of it. So yeah,
and
absolutely.
then we're swarming and resistant genes.
Yeah, right. So yeah, so the use of manure, you guys, is really, they're trying to see if they can't control the use of manure in food crops more. Yeah. Or, because now they're starting to be worried about this DNA contamination, not just the bacterial contamination, it's the DNA contamination. And through horizontal gene transformation, those antibiotic resistance genes could be taken up by the animals, normal microbiota by our normal microbiota. Yeah, it's a hot topic, big topic. Yeah. So again, you guys, if we could just reduce the use of antibiotics in agriculture, that would be a big start. That'd be a huge start for us. Okay, sorry, that's kind of like. Now, just so you all know, in the PowerPoint, there's additional slides, but we're going to cut off the information for saying right at this point. Okay. All right. So, thank you. Okay. So, as I mentioned earlier, guys, PCR and gene cloning in your handout, page 2 and 3. Those topics are scientist homework. I tried to give you some reference pages here, so refer to page 252 for PCR, and then general reading assignment for chapter 9, pages 246 to 252, and there's also a clinical, pages 246 to 252, and there's a little forensic microbiology tidbit on pages 262 to 264. And there's a clinical focus on how PCR is used to track outbreaks of pathogens on page 266. So that would be your homework assignment out of chapter 9. And again, the practice quiz on D2L, that will help you with some of the specifics of PCR and gene cloning. Right. And then attached, you guys, at the very end, this is a little bit dated. It's from 2007. But this guy, Stuart, he's one of the leading science proponents of controlling the overuse of antibiotics. And again, even though this article is a little bit old, I think it's one of the best ones I've read on antibiotic resistance. So hopefully this will help reinforce some of the topics we've discussed here. Okie dokie. So with that, you guys, we're going to move to chapter 13. And we'll just see how far we can get. So viruses, you guys, they're scary and fascinating. Yeah, they're just, oh my goodness. And the more folks learn, the more biologists learn about viruses. One of the things, and I don't talk about this, is viruses are probably a powerful force in helping to generate genetic diversity. They probably played a scientists are just starting probably to really appreciate that. So unfortunately, all I'm gonna do is kind of focus on how they kill things. But just have that in the back of your mind that these viruses may have played a really important role in the evolution of genetic diversity here on Earth. And maybe, and we'll talk about some ways, maybe we can, as we learn more about viruses, maybe we can learn ways that we can use them to help as well as to harm. All right, so I love this opening. So a famous scientist was asked, what are viruses? And he replies, viruses are trouble wrapped in protein. And I thought, that's a great way to open this. Okay, so viruses, trouble wrapped in protein. So we're going to talk about viral history from a human perspective, how we see viruses. So first of all, they're really important to us because they caused many of the great plagues throughout history. So, for example, viruses cause smallpox, viruses cause influenza. So millions and millions of people have died as a result of these viral infections over the ages. And one of the problems was, since viruses are so small, they're much smaller than bacteria, you can't use a light microscope to visualize most viruses, so they remained a mystery for much longer than bacteria did. And that caused a lot of probably additional loss of life and sorrow, because nobody knew what was causing these diseases. So it was in the 1800s that some folks started thinking maybe there was an infectious disease agent smaller than bacteria, but they just didn't know what the heck it was. So the first really important work, and it made me sad because in this book by Nathan Wolff, Ivanowski wasn't given credit, and I don't think that's right. Dmitry Ivanowski in Russia really did the first crucial work dealing with viruses, and I think he should be called the father of virology. In Russia in 1892, Dmitry Ivanowski was trying to figure out what caused the disease of tobacco plants called tobacco mosaic disease. So here we see a tobacco leaf that's been infected, suffering from tobacco mosaic disease. And you can see these little splotches of discoloration. So folks said it almost looked like little mosaic tiles, these little light discolored spots. In contrast, here is a healthy tobacco leaf. And you all know that for many, many ages, tobacco has been used as for smoking, for chewing, right? So it's of economic value, even though now we know it causes all kinds of cancer, so don't use it, guys. All right. So what Ivanowski was doing and trying to figure out what the heck was causing the tobacco mosaic disease, he was using these beautiful porcelain filters that had been developed at the Pasteur Institute. And these filters were used to filter out bacteria from solutions. So these porcelain filters, they had tiny pores or holes through which bacteria can't pass. Bacteria were too big to pass through them. So we'll just cartoon this, you guys. So let's say this is our filter. This is our ceramic filter. And the filters have these little tiny pores, so small that if you had a solution, so this is a solution, and let's say it had bacteria in it. So these are our bacteria. These are the pores. The bacteria were so big, they couldn't pass through the filter, so they'd be caught on the filter. The fluid, however, could get through. So your filtrate then would be bacteria-free. There'd be no bacteria present. Now, these porcelain filters were used in experiments to try to figure out what was causing a disease. So Ivanowski's experiment was to take leaves from a tobacco plant suffering from tobacco mosaic disease. He would grind them up. Okay, so he would grind up the leaves, the T tobacco mosaic disease leaf. Okay, he would mix it with water, sterile saline, and then he would filter this commish through one of these porcelain filters. Okay? Now the idea was that if bacteria were responsible for causing tobacco mosaic disease, they would get caught, in the filter. The filtrate would be bacteria-free. He would then use the filtrate. Filtrate was used to inoculate healthy tobacco plants. And if indeed the cause of the tobacco mosaic disease was a bacterium, since the filtrate wouldn't have any bacteria in it, when he inoculated healthy tobacco plants, he would predict the plants would not become diseased, right? If bacteria were the cause. But the surprise result was when Ivanowski did this experiment, got his filtrate, he inoculated healthy tobacco plants. What do you think happened? These became infected. These developed. The surprise result was that the plants developed tobacco mosaic disease. And so what Ivanowski proposed was that there is some infectious disease agent, and I'll just put like little tiny stars here, some infectious disease agent smaller than bacteria that were able to pass through the pores. So they were present in the filtrate. So when you use a filtrate to inoculate healthy plants, that infectious disease agent was present, and that caused the tobacco mosaic disease. But they didn't know what it was, right? All the smallest thing that folks knew about were bacteria. So Ivanowski, in reporting his results, he just called it a filterable agent because it was able to pass through these bacterial porcelain filters, right? So as you guys know in science, it's so important when you do an experiment and you get, really, incredible results, it's important that somebody else repeats your experiment and gets the same results. So later, Martinus by Gering, a Dutch botanist, repeated Ivanowski's work, and he got the exact same results, which is dynamite. Now, Martinus by Gering, I was just reading in the Nathan Wolf's book. His father apparently had been a tobacco merchant, and tobacco mosaic disease had wiped out all the tobacco plants, and by Gering's father had become bankrupt, so he had kind of a passion for this. So he repeated the result, and by Gering used the term for the it. Instead of calling it the filterable agent, he called it the filterable virus. And virus comes from the Latin for poison. And what by Gering thought that this filterable virus, he thought it was soluble, like a poison. That's why he calls it a poison. But because by Gering was the first person to use the term virus, he gets claimed as being the father of virology. And no doubt, he's really important, but I really think Ivanowski should have been given that. In scientific circles, you guys, it's just like everywhere else in society. There's egos and, little intrigues, a little backstabbing going on for that. So, this is a big deal in virology. Who do you think should be the father of virology? Ivanowski or by Gering? Well, maybe they could be co-fathers, right? They could be co-fathers. All right. So, we'll keep going here. So then we're going to fast forward to, a very sad time in world history, World War I, 1914 and 1918. And it's so sad, ironic. Sometimes major advances in medicine are made during warfare because all this money is going into, how to prevent the troops from getting ill, or sometimes major advances in surgery. So outside Paris, there was a Canadian bacteriologist that was trying to investigate why a bunch of the cavalry were developing dysentery diarrhea. And just like you guys would do if you were working in a microbiology lab, you would get samples of the sick patient's feces, and just like you guys do in micro lab, you would inoculate agar plates with the fecal samples so you could see if you could identify maybe some pathogenic bacteria growing, maybe salmonella or shigella, maybe Vibrio cholera, right? So what the bacteriologist observed, and I'll have to explain this a little bit, you guys. As you all know, sometimes if you have lots and lots of bacteria, they'll make a lawn over the surface of your plate. We call it confluent growth, or all of the colonies touch one another. So it looks like a lawn, right? So in the experiments, they would get these lawns of bacteria, but after incubation, they would hold the plates up to the light, and they would see little holes in the lawn of bacteria where the colonies weren't growing, where it appeared that bacteria had been destroyed or eaten. These little holes in the lawn of bacteria, they have a fancy name, they're called plaques. And So at the time, again, nobody knew what viruses were. The researcher, Dave Harrell, said, you know, these little holes in the bacterial lawn, it was caused by a bacterial eater, a bacteriophage. Okay, a bacteriophage literally means bacteria eater. So again, they didn't know what these bacteriophages were, but they thought, okay, somehow they're destroying the bacteria. We now know these bacteriophages were viruses that were infecting and killing the bacteria. But again, back in the early 1900s, nobody knew who these bacteriophages were. We still use this name, you guys. We still use the term bacteriophage for viruses that infect bacteria. Okay, so of great interest was that Dave Harrell suggested, could these bacteriophages be used to kill pathogenic bacteria? And this concept was worked on a great deal in the former Soviet Union prior to World War II. You guys remember in World War II, that's when penicillin came into its own, and following World War II, many different antibiotics were discovered. So when antibiotics were discovered, this interest in using bacteriophage to kill pathogenic bacteria fell out of favor. Everybody was like, oh, the antibiotics will kill them. But now that we have all these antibiotic resistant bacteria, now a lot of research is starting again to see if bacteriophages can be used to kill antibiotic resistant bacteria. So, you know, it's like history is important. We learn from history, and history often repeats itself. Okay, so again, observation of the effect of viruses on bacteria. But again, nobody knew these were viruses. So we fast forward to 1935, UC Berkeley here in California, and a biochemist by the name of Wendell Stanley took the tobacco mosaic agents, and he was able to crystallize it. And this had people so confused, because to be able to make crystals out of the material, usually that's a property of chemicals, not of cells. So people were really confused. So they thought, well, maybe these viruses, maybe they're all made of protein. But then a little bit later, some additional research over in Britain by another biochemist discovered that the viruses were primarily protein, but there was a little bit of nucleic acid present. So they refined their description and said viruses are nucleoprotein, nucleic acid and protein. So again, but they have no idea what they look like. All right, now again, you know, what was the problem here? What was the holdup? If we look at the size of viruses, they're down here in the nanometer size range. Just so y'all know, and this depends on the textbook you read, but virus size range, in general, what I always remember, it's around 30 nanometers, which is about the size of a ribosome. The polioviruses are around 30 nanometers, all the way up to about 300 nanometers. And this is the largest, or some of the largest viruses, the pox viruses. They now have a larger virus called the Mimi virus, so I should put that in here. But the Mimi virus is truly an unusual virus, relatively new discovery. And it's around 600 nanometers. Okay, but this is just like a G whiz for us. This is a G whiz. So in general, most viruses fall between 30 to 40 nanometers to 300 to 400 nanometer size range. Now, the problem is that it's only these largest viruses that we just might be able to see with our light microscopes. Most viruses are too small. We can't see them with our light microscope. So this was what was, hindering understanding of viruses. As humans, we want to see them, right? We want to see what their structure is, and if we know the structure, maybe we can figure out the functions. So it took the invention of the electron microscope, which permits far greater magnification than light microscopes. The electron microscope was first developed in Germany in 1932. The first electron microscope was built in the United States in 1939 by RCA. So a group of scientists that were interested in understanding viruses got together, and they were going to use the electron microscope to take a photo, take an image of bacterial phages. And these guys, they were so excited. Nobody had ever seen a bacteriophage before. So when the image emerged, they were just awestruck. And one of the researchers from Europe, he said, "My God, they've got tails! My God, they've got tails!" And so they were just blown away. And just imagine you guys, seeing this image emerge. You're like, oh, come on, this is some, dorky science fiction movie, right? But no, this is an image of one of the bacteriophages. It truly looks like some alien, right? So the scientists that were awestruck, they were trying to describe it. They say, well, they look sperm-like because they have little tails. They have tadpole-like. To me, it looks like a little lunar lander or something right out of Alien, And I've always said, you guys would be the best folks writing science fiction stories in Hollywood, because there's no greater source of great science fiction than nature, right? Okay, so this is indeed an image of a bacteriophage. This is one of a group called the T-phages, and what we're seeing here are protein structures. And just really quick, and we'll go into structure more, this is called the capsid, or the head. And again, it's a protein shell, and inside is the nucleic acid of the virus. Okay. So this is why that first researcher said that viruses are trouble, meaning the genetic information wrapped in protein. Okay. So we'll come back to a little bit more on history later. This was one of the great researchers in virology. When asked to describe, what it was like to be a virologist to study these bacterial viruses, he said, it's like a fine playground for serious children to ask ambitious questions. It's like, it can be fun. Like kids have fun exploring their world, and it's like some of these areas, it's like, oh, if we could just maintain that childlike wonder, it's just like, how much fun is this? Okay, a little bit on vocabulary, folks. I'm very bad. I'm a bad teacher, because I don't distinguish between virus and virion. In lecture, I just use the term virus all the time. So we have a description of a virus as a tiny infectious acellular agent with nucleic acid surrounded by proteinaceous capsamers that form a covering called the capsid. So that's the description of the virus. But you will see in your textbooks the term virion, and virion is the term used to describe a virus which is outside of its host cell. We're going to see viruses have to be inside a host cell to replicate. When the viruses are outside their host cell, officially I should call them virions. They're not replicating, they're just waiting to invade a host cell. But as I said in lecture, I almost always use the term virus, so I just wanted you to be aware of that. So general characteristics of viruses, and reference here in your textbook is page 378. So if I were to ask you to describe some of the properties, some of the traits of viruses, this would be a partial list. So remember, you guys, they're acellular, they're not made of cells, they don't synthesize a cell membrane. That's the key to being a cell. Can you synthesize a cell membrane? If the answer is no, you're acellular. Viruses can't synthesize cell membranes. Viruses lack ribosomes. That's another reason why they have to invade a host cell. They have to get the host cell ribosomes to make the viral proteins. They don't carry out metabolism. They can't generate their own ATP, so they have to steal ATP for biosynthesis from the host cell. This is very cool, you guys. Viruses have either DNA or RNA, not both. In contrast, cells always have DNA as their genetic information, and cells also have RNA. Viruses are classified either as DNA viruses if they have DNA genomes, or RNA viruses if they have RNA genomes. Viruses don't grow big and then divide, as we see in cells. Instead, viruses are assembled from parts, like you'd assemble a car. Viruses can only replicate, that is, make copies of themselves if they invade a host cell. So they have to use all the biosynthetic machinery of the host cell to replicate. And hence they're called obligate intracellular parasites. This is just page 384, table 13.1. This is just a table that helps you compare the properties of viruses to the properties of cells. So maybe it might be a helpful study guide. So what we're first going to do is look at the structure of viruses, and there's three basic types, and we'll focus primarily on the first two types. The so-called naked viruses, or non-envelope viruses. These are viruses where the outermost layer is that protein coat. But then we'll see that some viruses, when they are escaping from the host cell in which they've been replicated, they'll steal some of our cell membrane. So it might be they'll steal some of our cytoplasmic membrane, they might steal some of our nuclear membrane. That membranous coat is referred to as the virus envelope. Okay, and so these are called envelope viruses. Now before they steal our cytoplasmic membrane or nuclear membrane, viruses always insert some of their proteins into that cell membrane. So we'll always have viral proteins in the envelope. And then the third group are the complex viruses. We'll have some pictures of some complex viruses here. They're, just as their name suggests, they have a little bit more complicated structure than these other guys. So we'll see some pictures. So this is a cartoon, figure 1311 on page 278, of kind of a generic naked virus. And again, by naked we mean the outermost layer is a protein coat. This protein coat is referred to as the capsid. And the capsid is made up of little subunits called capsomeres. And the capsomeres can be organized into different shapes. And we'll see that two of the most common shapes are polyhedral shape and helical shape. And again, we'll come back and see some photos of that. The most important job of the capsid is to protect the viral genetic information inside. And again, the viral genetic information, it could be DNA or it could be RNA. Now, in naked viruses, another important function of the capsid is it will have special proteins called adhesins. And adhesin sounds like adhesive tape, and that really describes their functions. The viral adhesins will bind to complementary molecule on the surface of the cell they're going to invade. So the viral adhesins are essential for the virus to attach to the surface of cells. This is very important, you guys. Since naked viruses, since the adhesins are associated with this tough, strong protein coat, naked viruses can remain infectious outside their host for long periods of time, and that's bad news for us. If we have naked viruses that are causing outbreaks of disease, we need to remember the environment can become contaminated. The environment can act as a source of those viruses, so that's bad news if we're trying to control an outbreak of disease. And one example of a naked virus are polioviruses, which are shed in feces. So that means that once the polioviruses are shed to the host in feces, wherever the feces goes, the polioviruses go. So they can contaminate drinking water, they can contaminate food. And another example of a naked virus are the papillomaviruses, the so-called wart viruses. They too can remain infectious in the environment for long periods of time. These are just examples of some diseases caused by naked viruses. This is a photo from around the 1950s with the horrific polio outbreaks. This was prior to the introduction of the Sabin and salt polio vaccines. So sadly, in some folks, the polio virus makes its way to the central nervous system and can cause damage that will result in paralysis of the respiratory muscles. So folks that had those polio infections, they would be kept in these iron lungs that use positive and negative pressure to help folks respire. Yeah.
I have a question. It's not a little bit
Yeah.
off topic, but I was reading that like the smallpox vaccine, they were talking about like giving the vaccine to everyone, how it would kill a lot of people, just giving them the vaccine. But I know with my son got the polio vaccine when he was 2 months old, but does a polio vaccine ever harm kids accidentally?
Well, actually, and we will come back to this, but just in a nutshell, there's two different polio vaccines. The Salk vaccine, which is a kill virus vaccine that you inject, and that will not cause polio. But the second polio vaccine, which was used here for quite a while in the United States, was the Sabin oral polio vaccine, and that's a live attenuated virus. It means it's been weakened, and in most people it won't cause polio, but sadly it sometimes reverts to virulence. So you can have polio caused by the Sabin polio vaccine, and that's why here in the United States we've switched from the Sabin oral polio vaccine back to the Salk injectable, because we don't want to risk causing polio in any of our children to be vaccinated. It's a great question. We are going to come back and we'll have like a whole little section on that whole vaccine debate, and there's some other issues that are involved. But it's a great question. Yeah. So, and we'll talk about the difference between killed virus vaccines and attenuated live virus vaccines and who you want to give each one to. Yeah, good question. Okay, and then another example of a naked virus that can cause infection, the papilloma or wart viruses. We will come back and talk about these different viral pathogens, guys. So different examples of warts, again, bad news. Naked viruses remain infectious in the environment for long periods of time. is one of the viruses that can cause relatively mild upper respiratory tract infections, which we call colds. And one of the cold viruses is the rhinovirus. It too is naked, and that helps us understand why some of the cold viruses remain infectious outside of the host for long periods of time. The second group we're going to talk about are the envelope viruses. And you guys remember, envelope viruses steal some of the host cell membrane before they escape. It could be cytoplasmic membrane, nuclear membrane, it could be membrane of the endoplasmic reticulum, it could be membrane from the Golgi. And usually we use cytoplasmic membrane as our example. So this represents the envelope, the stolen host cell membrane. And again, you guys want to know, the virus has to insert proteins into the envelope. And one of the important functions of some of these proteins will be to act as adhesants. Inside would be the protein coat or capsid. Here we have a different shaped capsid. This is a helical capsid. And the little red ribbon here, that represents the genetic information of the virus. Now, let me just take a look here. The good news about envelope viruses is since the adhesins are in the envelope, and because the envelope is very delicate, you guys remember when we described the fluid mosaic model of cell membranes, we said that phospholipid bilayer has the consistency of olive oil. So consequently, when envelope viruses are shed into the environment, that envelope is easily destroyed by drying, by dehydration, by sunlight, by soap, by detergents. Mild chemicals will destroy that envelope. And consequently, that means that envelope viruses usually do not remain infectious in the environment for long periods. And we are so lucky, you guys. One of the few good things about HIV, it's an envelope virus. Not going to remain infectious in the environment for long. Okay, influenza virus, envelope virus. So thank heaven some of these, really, really virulent pathogens are enveloped. And that means they will not remain infectious in the environment for long. Thank heavens. Whew. Here's some examples of some envelope viruses. As we mentioned, HIV, this electron micrograph of HIV budding from an infected human cell, and it steals part of our cytoplasmic membrane with it. And again, the good news is, you guys, that envelope is easily destroyed, so we don't have to worry about catching HIV from some inanimate object. The herpes viruses, which we'll also be talking about, there's a great variety of herpes viruses. The good news is they're all enveloped, so again, they're not going to remain infectious in the environment for long. This is just to give us an idea of the size range. And again, you guys, depending on the textbook you read, you'll get different size ranges. I always like to remember 3 nanometers or 300 nanometers. It's easiest for me to remember. And this just gives us an idea of size comparison. Human erythrocyte around 7.5 micrometers to 8 micrometers. Bacterium around 1 micrometer. So our viruses are around 100 times smaller than a bacteria, or around 1,000 times smaller than a blood cell. This is figure 13.4 on page 381. And again, just to zoom in on those viruses, here's our bacterial cell wall. Bacterial ribosomes around 25 to 30 nanometers, and we can see a lot of our viruses are about the size of a ribosome. Colovirus at 30 nanometers. Bacteriophage here around 20 to 50, up to 225 nanometers. Tobacco mosaic virus, a lumpy helical virus, 15 nanometers by 300. And here's one of the larger viral families of pox viruses. Here's a smallpox virus around 200 by 300 nanometers. We might just barely be able to see smallpox virus on the light microscope. So what we'll do then, you guys, is after the break, we will continue our discussion of viral structure, and then, you guys, if you have time, try to read about bacteriophage replication. The table